This is the pre-lab and instructions for lab number 7, gel electrophoresis. This is the lab that we would have performed right after spring break in order to separate the PCR products that we had amplified the week before spring break. So what exactly is DNA gel electrophoresis? Simply, gel electrophoresis is just the technique used to separate DNA fragments according to their size. This next slide is kind of a wall of text, and you can refer to it to help you with some of the questions for the assignment. But the principle of gel electrophoresis is that DNA samples are loaded into wells or indentations at one end of a gel. The gel is made up of agarose, which is kind of a gelatin-like substance. An electric current is applied to them and it's pulled through the gel. The two reasons that DNA gel electrophoresis works is because DNA fragments are slightly negatively charged. So if you apply a positive current or a positive charge, they're going to be attracted to that positive charge. The other reason that it works is because if you have a semi-solid or a gel solution, fragments that are smaller are going to be sleeker and more streamlined, and they're going to be able to move more quickly through a thicker substance. In much the same way that a thinner, sleeker swimmer is going to be able to move through the water much more quickly or with much more ease than somebody that's a lot larger. So it's for those two reasons, the negative charge and because smaller fragments will travel faster than bigger ones, that this technique works to separate things according to size. The other thing that's in the gel is that the gel is stained with a DNA binding dye that fluoresces under UV light so that you can actually visualize where the DNA has migrated to in the gel. So after that, an example, this is kind of a stylized version of an agarose gel that has been run. And what you see is typically there is some sort of a marker on the left-hand side. And these are known sizes of DNA fragments so that you have something to compare your unknowns to. In this case, we know that the sizes of these range from anywhere between 1,200 base pairs and 100 base pairs in 100 base pair increments. From there, we can see where the samples line up. So sample A lines up with the 1,000 base pair band. So we know that that A is about 1,000 base pairs. Sometimes if you have a pool of different sized samples, this is important. It will separate these out so that you can evaluate the size of 2 or 3 or even more bands. So in the case of B and C, there are more than 2 sizes of fragments of DNA in the samples. So why are we doing this exactly? So the whole point of this lab is to be able to determine our genotype, whether we have the PV92 allele insertion or we don't. And if it's on one chromosome, both chromosomes, or some combination thereof. So in lab number 5, we extracted our DNA. That was the part where we rinsed with a saline solution, spit it into a cup, centrifuged it down a bunch of times in order to get a cell pellet, and extracted the DNA. Lab number 6, we figured out how much DNA was in that sample, like the physical quantity in nanograms per microliter. And then we took a small sample of our DNA and subjected it to PCR. And what PCR is doing is it's photocopying just that region of interest that we're looking at. So essentially what PCR is doing is it's taking the pool of all of your DNA all 23 pairs of chromosomes, and it's honing in on chromosome 16. And specifically, you have 2 chromosome 16s, one from your mother, one from your father. And it's looking at that specific region on chromosome 16 to determine whether or not you have that ALU insertion in this particular region. If you have the ALU insertion, like the one on the left, then your chromosome 16 is technically 300 base pairs longer than a chromosome that does not have it on there. So what PCR is doing is it's photocopying the region where the ALU insertion is, plus a little bit upstream and downstream of where that insertion is. If you have the insertion, it will be approximately 941 base pairs, which you could determine by just looking at the size in comparison to the ladder. If it's ALU negative, then it's going to be 300 base pairs shorter, or about 641 base pairs. And if you have 1 copy of each one, then you're going to get 2 bands, one at 941, one at 641. The reason that these are upstream and downstream, we want to kind of photocopy the region around it, is to determine whether, if no band appears, whether it was just because you don't have the ALU insertion, or whether it was because there was something wrong with your sample. So even if you don't have the ALU insertion, you should still have a band. Versus if we just tested for this ALU insertion, and if you had it, you just have this 1 band, you'd only be able to tell whether you had it, not whether there was something wrong, or even whether you were heterozygous, meaning you had 1 copy of the ALU insertion and 1 chromosome without it. So after PCR, your tube contains DNA fragments in one of 3 scenarios. The top scenario is if you are positive, meaning you have both chromosomes that have this PV92 insertion. You are heterozygous, meaning that one of your chromosomes has the ALU insertion, the other one does not. So there's 2 different fragments there. Or whether you're negative, and neither of your chromosomes has that PV92 ALU insertion. So because these guys are different sizes, you need to separate them out on the agarose gel in order to evaluate the size, and also to determine whether you have 1 band, 2 bands, whether you're homozygous or heterozygous. So what we would have done is, after you retrieved your tube, you have 40 microliters total volume in your tube. 10 of that was DNA, or 20 of that was DNA, 20 of it was that PCR master mix. We add a loading dye to the sample. And the PCR sample kind of had a yellowish tint to it, but it's pretty faint. The loading dye serves 2 purposes. The first one is so that we can track it as it migrates through the gel. So it kind of gives it a little bit of a color. The second reason is that the density of the liquid in your PCR is the same density as the liquid in the gel electrophoresis apparatus. So if you try to pipette your sample into one of these wells, it would just float up and out of the well. So the loading dye has a little bit of glycerol to weigh the sample down so that it stays in these wells until it can be pulled into the gel. So you add 10 microliters of loading dye, and from there, each one of these wells holds about 20 microliters. And this is a video showing how the DNA gel is loaded. See, Daniel in the lab. See? You got it, dude. Here? Mm-hmm. You feel it? Huh? Yep, and push it down.
It's not going in.
Here you go, and then while you're holding it down, pull it up. There, see? You did it. It was awesome. See. So that was just an example of how one of the wells on the agarose gel is loaded. So you'll notice that the agarose gel is kind of a slab, like a gelatin slab, and it's also in the gel box where the electrophoresis takes place. There's a buffer that keeps it, it's kind of an electrolyte buffer that helps to carry the current, but also kind of keeps the temperature down as well. So after the samples are loaded into the gel, an electrical current is applied. And if you can imagine that the top of the gel is the part of the gel that's furthest away, and the samples are running downwards or towards you where the electrical current would be. So when you apply the electrical current, the samples start to migrate, and you can see that the faster ones are the smaller fragments. So the 100 base pair fragment falls into line towards the bottom, and the 1000 base pair fragment falls in line towards the top. You can see here that the fragments in the positive, this lands at about 941 base pairs. There are 2 fragments in the positive, and there's 1 fragment at about 641 in the negative. From there, you can determine what the genotype of sample S1 and S2 is, either by comparing it to the ladder sizes or by looking to see whether it matches one of the control scenarios. So S1 has the genotype positive, negative, while sample S2 has the genotype of negative, since it matches the negative pattern here. So the way that we load the samples into the gel, ordinarily we do have a ladder. It hasn't been working lately, so we haven't loaded it in there. Lane number 1 instead has a positive control sample. This is a sample that is known to be positive. And if it didn't work, then we would start to question whether something was wrong with our reagents or our equipment. The second one is positive, and the third one is a negative. So we have all 3 different scenarios. Typically we skip a lane just to provide a little bit of space between the controls and the next sample. And then 20 microliters of student number 1's DNA, number 2, number 3, and number 4. So when the gel is loaded, you'll have all the samples in the top. And as the electric current is applied, you can see all of the samples starting to migrate towards the red side or the bottom of the gel. So this is after about 15 minutes. This is after about 30 minutes. I let it go for 45 minutes to an hour, and the yellow or the orange color is almost all the way towards the bottom here. If you're just running the control samples, what you might see is the bands have separated out. So this one is the positive, and it runs towards the top of where you loaded the sample. Positive, negative produces 2 bands, one that should be about the same size as the positive, and the other one that should be about the positive or the negative. So 2 bands, the same size, as either of the other ones. And then in lane number 3, this is the negative negative. There is a lot of DNA in these wells. That's why it appears like the image is blown out a little bit. You want to kind of look at the very bottom, or kind of the middle bottom of each lane in order to figure out what the size is. So in this case, it does look like it's a little bit larger, but that's just because there's a lot of DNA there. And here it's blown out all the way up. So it looks like it's a lot bigger than it actually is. So for our class, and this is part of the assignment, is analyzing our class gel. And this is an example from one of the sections, but it'll give you an idea of what you need to do. So in lanes 1, 2, and 3, this is the positive, 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 negative, negative, negative. For each sample indicated, determine what the genotype of each student was. So lane number 4 is empty. Lane number 5 did have something in it, but we can't see whether there's a band there or not. So that one's indeterminate. Lane number 6 has a band that lines up with this bottom band, and there's only one, so it's negative, negative. The next lane over also had DNA in it, can't be determined. The next one over is negative, negative as well. It lines up with that bottom one, and so on down the line. Now these look like they could have been positive, negative, but the band up above is not quite as bright. So these are also negative, negative. This one here is a positive, positive. I've also included images, aside from being labeled, they also have lines to kind of help you line up where the bands are. So you can see whether it's actually a positive, positive, or it's actually negative. So to complete in Canvas, there is an assignment in the form of a quiz that is under lab number 7. It includes questions about why gel electrophoresis works, why we're performing gel electrophoresis, and then analyzing the gels from our lab. This can be completed at any time. The sooner the better, the sooner that you'll receive a grade after it's been submitted. But please, again, do it before the end of class, before the end date of class.